In this short video, we're going to look at the structure of viruses. So remember that viruses are made out of two things, a nucleic acid and a protein coat called a capsid. So with the nucleic acid of viruses, they could use either DNA or RNA as their genetic material. And some viruses use double-stranded DNA, others will use single-stranded DNA. Some use double-stranded RNA and others will use single-stranded RNA. So how is this different from cells? So remember that cells will only use double-stranded DNA as their genetic material. They will never ever use any of the other three. So that's what's really different with viruses compared to cells. So the protein coat, called a capsid, is made out of subunits called capsomeres. Remember, mere means subunit or part. The capsid is actually very easy to put together. Once the pieces are made, the capsomeres, they will just self-assemble. The functions of the capsid, there are two of them. The first one is that the capsid will protect the virus's nucleic acid. And then the second function is that the capsid has some proteins on the surface, like you can see in this picture over here on the right of the adenovirus, which is one of the viruses that causes the common cold. So you can see that these proteins that are sticking out of the capsid, they're actually used to bind to receptors on our cells, and it tricks our cells into letting them inside. There are three different shapes to the capsids. There are helical capsids, icosahedral capsids, and then complex capsids are just ones that don't fit into either helical or icosahedral, so they're just called complex. Here's a picture of a helical capsid. So you can see that it forms this spiral hollow tube, and inside the viral nucleic acid is placed. So this is actually an electron microscope view of the Ebola virus, and you can see its helical capsid here. And it does not have an envelope. Here is the influenza virus. So you can see inside we've got the envelope here on the outside in yellow, and on the inside we've got the blue capsid that's made out of the protein. And you can see how the capsid is helical in shape, and you can see the nucleic acid that's inside. Here's the adenovirus. It has a capsid that is icosahedral in shape. Icos means 20. And hedral means faces. And it's just talking about the fact that there are 20 triangular faces that are put together to make up the icosahedral capsid. Complex capsids are the third shape that capsids can be, and anything that does not fit into a helical capsid or a icosahedral capsid, we'll call them a complex capsid. So here on top, we've got a picture of a bacteriophage, or a virus that infects bacteria. And you can see that the head here actually has an icosahedral shape, but then the sheath here is helical shape, and then it's got these tail fibers coming off. So it really doesn't fit into completely helical or completely icosahedral, so to be a complex capsid. And then on the bottom, we have an envelope virus. You can see the envelope on the outside. And then on the inside, you can see the capsid. So that capsid is not helical, it's not icosahedral, it's kind of a dumbbell shaped. So this one would also have a complex capsid. And this is the smallpox virus. So some viruses have an envelope, but others do not. So if they do not have an envelope, they're called non-enveloped or naked viruses. So you can see on this picture here on the bottom, you can see the envelope that's on the outside here. The envelope is made out of three things. So if you look closely, you can see there's a phospholipid bilayer. So one component of the envelope that it's made out of is phospholipids. There's also some carbohydrates, and the third thing that the envelope is made out of are proteins. So this influenza virus, you can see all these spike proteins sticking out, and that would be the third component of the envelope. So envelopes are made out of phospholipids, carbohydrates, and proteins. How the envelope is made? The envelope actually comes from the host cell. So as the envelope virus leaves the cell, it will pick up either part of the cell membrane, or it will pick up maybe one of the membranes of one of the internal organelles, like maybe the Golgi apparatus. And so then as it leaves, it takes that envelope with it. The only part of the envelope that the virus actually makes are the spike proteins that are sticking out here, and then everything else is made by the host cell. So the phospholipid bilayer and carbohydrates come from the host cell. The function of the viral envelope is to bind to the host cell. So if you look on the top right here, you can see a picture of the influenza virus, which has an envelope here in blue, and sticking out of the envelope are all the spike proteins that are made by the virus. So some of these spike proteins that the influenza virus makes that are on the envelope will actually bind to receptors on our cells, and that's how the influenza virus gets inside of our cells. The big disadvantage to having an envelope is the envelopes are very fragile. So they can dry out very quickly, and then once they do that, they disintegrate, or they can be disrupted very easily with soap and water, alcohol, disinfectants. So there's a lot of things that can destroy the envelope. Once the envelope disappears and is gone, all of the viral spike proteins are now gone as well. So the only thing left would be the capsid with the nucleic acid inside. And because these envelope viruses had put their spike proteins on the outside in the envelope, they can no longer bind to the host cell, and so they're no longer able to infect cells, and so they're pretty much dead, and so there's nothing that they can do. If you're interested, there's a link here to an article that talks about how long cold and flu viruses stay contagious on surfaces. So it's kind of an interesting article to read. And I'm sure you've been hearing about the coronavirus as well. It's an envelope virus, and they've done some studies, and they found that it can survive on cardboard for up to 24 hours and still remain infectious, and then on plastics and metal for 2 to 3 days. And then after that time period, the envelope has just disintegrated. There's no more spike proteins. It can't bind to your host cells anymore and get inside, and so it's no longer infectious. So the viral envelope proteins, these spike proteins, we've kind of mentioned these already, but there's two functions that they can have. So on our influenza virus here, you can see that there's two different types of spike proteins. So the hemagglutinin spike protein that's in yellow, that's the one that's going to bind to the receptor on the host cell and then allow the influenza virus to get inside of our cells. So other spike proteins can act as enzymes. So these green spike proteins on the influenza virus, they're actually an enzyme. And the enzyme is called neuraminidase. And remember we talked about that before when we talked about the drug Tamiflu. So what the neuraminidase enzyme does is that when the flu virus is leaving the host cell and it's taking part of the host cell membrane with it, that neuraminidase enzyme will cut the anchor and release the virus from the cell, and then it can go and infect other cells. So Tamiflu actually blocks that neuraminidase enzyme. So with the flu virus, you probably have heard of different names for it. There's different strains of it. So you might have heard of like the H1N1, H3N2 influenza viruses. They're just named after the type of hemagglutinin and neuraminidase proteins that that particular flu strain has. So that's where the names come from. Okay, and then as we talked about, some viruses do have enzymes. They're not going to be active until they get inside the host cell. A lot of viruses don't have any enzymes that they bring with them. They just end up using all of the enzymes from the host cell to multiply and reproduce. But there are some viruses that have to bring their own enzymes with them because our cells don't have them, so like influenza with the neuraminidase. And then HIV will bring in its enzyme reverse transcriptase because we don't have the enzyme to do reverse transcription.